Okay, so I guess everyone is doing well. And yeah, unlike me, yeah, actually I'm doing well naman as of the moment. Okay, so let's start. Let's get the ball rolling. If there are participants, if there are attendees coming, so I'm so sorry I might not be able to accept them because I will be sharing my entire screen, which means I will temporarily log out from my my Google Meet page. So okay. So this morning we are going to discuss the molecular radiobiology. So ipakita ko iyo an moving the hook. Uh, um, yeah, that the part in which hook has a little sort of transformation so that we can you can appreciate in kahit papaano the transformation of his molecules in his body into something like a weird one. So that's a common mythology, that's a common myth of X-ray effects of radiation. So, wala pa namang ganong nangyayari that after radiation exposure, the person will become a mutant like the Hulk. Okay. Anyway, so let's get the ball rolling. Dami kong introduction. So, let's start. Okay, good morning, everyone. So, this is molecular radiobiology. Okay, so molecular in a sense that, of course, our human body, if we can still have a short of a recap, we are composed of five major molecules in the body, four of which are what we call the macromolecules. Diba? If you can still remember, we have proteins, we have nucleic acids, we have lipids, and we have carbohydrates. So four are macromolecules. Macromolecules in a sense that, again, they are composed of a complex a composition. The composition of that molecule is quite complex, masyadong mahaba. So the other one, kasi nga five molecules in the body, the other one is the water. Water is a simple structure, diba? It is only composed, yan, it is only composed of dalawang atoms. Okay, dalawang atoms ng hydrogen at isang atom ng oxygen, H2O. Okay, molecular radiobiology. So it says here that the effect of radiation of macromolecules is quite different from irradiation of water. Okay, so allow me to make some highlights on this one. Again, macromolecules versus water. So that's why we need to study this one for us to be able to have a distinct concept of these two. So um, when we talk about water, isa lang ang papasok sa ating concept, especially when we talk about radiobiology. So if you already heard or read the word, ang bagal ko magsulat, radio, okay, radiolysis. So this is dedicated or radiobiologic response or pakiintindihin na lahat kung talagang pang grade 1 yan at nakasurat. Ako rin ngayon magsuratan ng computer. So radiolysis, this is dedicated, letter O ini siya ha, Pain pag chapter test nasugod. Sir, can you surat radiolysis? Radiolysis. So radiolysis, this is the radiation interaction with water, particularly in the human body. So again, the effect of irradiation of macromolecules is quite different from that of irradiation. So when macromolecules are irradiated in vitro, take note, again, we have the term in vitro. So bagaw naton may the letter T. Paborito nga letter ni Esquilio, letter T. So meaning this is with the test tube or outside the human body or outside the cell. So a considerable radiation dose is required to produce a considerable effect. Diba? So bago naton, bago sa PowerPoint, kailangan we should have a threshold. No, kung mag-i-study kita, effect of radiation to a particular molecule or a cell that is outside the human body, kailangan hataas muna ang radiation so that we can expect a radiation effect, di ba? Kailangan there should be a threshold dose to expect a response. In the other way around, irradiation in vivo, this is the opposite of in vitro, that is, within the living cell, demonstrates that macromolecules are considerably more radiosensitive, again, more radiosensitive in their natural state. So here, we can say that non-threshold pag natural natural state or in vivo. So nakuha naton. So halimbawa mag-thesis ka nga ni ngayon guys, if you have to do a research on, let's say, radiation effect versus a molecule, a substance of a molecule that is particularly a part of the human composition. So we can expect that there should be a higher dose if the experiment is conducted in vitro versus if the experiment is conducted in vivo. But again, we would like to clarify things. This is not to to establish fear about radiation. So remember, we have the 4R in radiobiology. 4R, Rongel Ristar de Maaram. So this is radiation, response, repopulate, and recovery. So kailangan we need to remember all of these things. Although human body or macromolecule is more radiosensitive in their natural state, but remember, the human body is so complex, so that we can really recover from a little amount of radiation. And take note, there's the concept of hormesis. A little amount of radiation is good for the human body. Okay, anyway, so let's establish that macromolecules, the effect of irradiation of macromolecules is different from that of water. Okay, so here we have, okay, the next slide says that a solution is a liquid that contains dissolved substances. I know for sure that when you were in high school, you mentioned about the three chemicals, yeah, sa chemistry. Kasi matagal na akong nag-high school eh. So I had my chemistry way back 2005 pa ata. So we have a definition of a substance, a solution, 'di ba? Ano pa 'yon? A colloid, yeah. So we have a different definition on these things. We need to be precise, especially sa chemistry. So a solution is a liquid that contains dissolved substances. So a mixture of fluids such as water and alcohol, also a solution. Okay, so when macromolecules are irradiated in solution in vitro, di ba parang mag-experiment ka ngayon, kailangan ihalo mo ini nga mga macromolecules sa isang solution. Yan. In vitro ha, meaning outside the human body. So based on the experiment, there are three major effects that happened. One is what they call the main chain scission. Scission, di ba? Parang ano yan, parang scissor. Diba? So parang ginugupit. No? Next, we have the cross-linking. Parang kumakabit. Wow. No, parang nagunting ka nga ni bond paper or paper, tas nagunting ka nga to nga something like ay didikit mo na liwat in another paper. So parang nagka-create ka ng cross-linking. So yung ginupit mong part, yung isang paper, nag-main chain scission ka, tapos ang the portion in which na-cut out mo siya, ipilit mo nga ni dito another paper, that is now another what we call the cross-linking. And we have point lesson. So here in our picture, we can say picture A is what we call the main chain scission. So let's take for example this green one. Itong parang bulate na to. Ewan ko ba kung bulate to. So this is, let's take this, let's consider this as like for example a protein. So we know that protein, the building blocks of protein is amino acids, di ba? And there are 22 amino acids in protein. So let's take for example ito, di na na-continue 1, 2, 3, 4. So Although 22 dapat yan, apat lang yung nakita kasi maulan nakita ang camera dire pausuan pag ano panorama. So halimbawang there is now this radiation, magi-interact siya, so matatanggal itong isa. So yung tinatawag nating main chain scission. So halimbawa, itong natanggal, dumikit siya dyan. So that's what we call a point lesion. And here, cross-linking. Yung isang natanggal, dumikit dito sa isang chain. So that's what we call another, again, cross-linking. So we can say letter B is cross-linking. And point lesion, if you can see, may mga asterisk dito. Asterisk ba yan? Or something like another structure. Okay, so we can say letter C is point lesion. Okay, so let's talk about these things in detail. In detail talaga. Okay, main chain scission. Again, tandaan natin yung gunting. Scissor. Diba? If you want to cut out something, like for example, let's take a bond paper as a protein. Ano? So, magka-cut out ka nga to kay gusto mo mag-produce ng art. So, yung ginunting mo, yung bond paper na ginunting mo, so parang it's a little bit a simple analogy of main chain scission. So when we say main chain scission, this is a breakage of a backbone of a long chain macromolecule. So ano talaga yan? Parang sinasabi dito na talagang si radiation, may guts siyang buwagin yung mga matitibay na pundasyon, yung mga matitibay na relasyon. Kaya sabi ng radiation na beater, walang forever. Hindi, may forever kasi later we will discuss that this is reversible. Okay, so again, main chain scission, this is the breakage of the backbone of long chain macromolecule. Favorite ko kasi si protein eh. Diba? Protein. So, let's take for example, this is protein. Again, si protein, meron siyang 22 amino acids. Diba? So, the result is a reduction of a long single molecule into many molecules, each of which may still be a macromolecule in a way. Like for example, so binuwag niya dito, side na to. Okay, left, beta, scission. So, ano siya, it is divided now into different fragments. Halimbawa dito, nabuwag niya. Wala talagang forever kay main chain scission. So, ito, mayroon ditong right beta scission. Yan. No? So, para ka lang nagugupit ng isang long chain molecule. So, guguntingin. So, yung gunting, yun yung radiation. Okay, so main chain scission reduces not only the size of the macromolecule, but also the viscosity of the solution. So, pag sinating viscosity, this is pagka malaki. Aray, pinapasok sa Google Meet. Sorry, later na lang pag YouTube. So, pag sinabi nating viscosity, this is the pagiging malapot ng isang solution. Yan, tulad ng tingin niya sa iyo na parang malapot na masyado. Ay, sana all. Okay, so halimbawa may sipon ka nga ni, makuri nga nito may gawas nga sipon. Ay, dapat may sipon yan na. Ako medyo makuri nga ni may gawas kay talagang masyadong malapot yan, highly viscous yan na solution. Pag halimbawa runny nose la siya, yan, medyo masyado malapot. Nagaano ako, may running nose. Nadalaga na niya erong. So that's lang siya, dire siya masyado malapot yan. For example, this illustration here, ayun. So ito kasi makuri mag-flow and fluid, so masyadong high viscosity, di ba? Versus dito na talagang tuloy-tuloy na lang, dumadaloy na. Parang pagmamahal niya sa'yo na parang dumadaloy na lang ng tuluyan. So this is low viscosity. So sabi dito, yung main chain scission reduces not only the size of the macromolecule but also the viscosity. So halimbawa, if this is the molecule, the protein that was initially without radiation, without irradiation, so, after the radiation effect, pwede na siyang maging ganito. Charin! Ayan. So, A viscous solution is one that is very thick and slow to flow, such as cold maple syrup. So tap water, on the other hand, has low viscosity. So measurements of viscosity determine the degree of main chain scission. So if we are going to illustrate this one, so parang, halimbawa, this is may na-irradiate na yung ano, na macromolecule, pero ganito lang niya naging effect. So masod ka nga, ay, di masyado ka severe and aton main chain scission. So yan. So the degree, pagka malapot ng solution determines the degree of, again, the degree of main chain scission. Now let's have the cross-linking. Parang continuation. If you can see here in the illustration, parang continuation siya. No? So some macromolecules have small spur-like side structures that extend off the main chain. Parang mga ganito. Yan, may tinatawag natin ditong cross-linker. So others produce these spurs as a consequence of irradiation. So case-to-case basis, ano, some meron na talaga cross-linker, parang ano na, nasa dugo na yung pagiging ano, mahilig mahilig ano, makikabit. Ay, wala namang pinapatamaan diyan na. At wala namang akong hugot diyan. Nasa dugo na ang ano, pagiging mahilig mga bit. So yung iba naman, ano, part lang or again, byproduct siya ng irradiation. Let's take for example, this one from the main chain scission, naging cross-linker siya. Ayun, so makita natin dito, meron dyan, mga cross-linker. Diba? So, kumakabit sa isang chain ng molecule. So, halimbawa from main chain scission, yung isang amino acid natanggal from the protein. So, pwede siyang, matatawag natin siyang magiging cross-linker siya pag dumikit siya sa another protein. So, hindi na, abnormal na yung protein, hindi na siya 22 amino acids, 23 amino acids na siya. So, yan, cross-linking yung tawag dyan. So, these side structures can behave as though they had a sticky substance on the end and they attach to neighboring molecule or to another segment of the same molecule. So, this process is called cross-linking, and radiation-induced molecular cross-linking increases the viscosity of a molecular solution. So, in a way, halimbawa, blood. Okay. So, after irradiation, mas naging malapot yung blood. Ano, mas nahirapan ng mag-flow because of the cross-linking. So, ganun. Halimbawa, isang chain ng molecule. Okay. So, mas naging malapot siya kasi there is now the presence of cross-linking. Diba? Nakikita nyo ba, if you can see the picture, medyo kaibahan siya kasi yung sa main chain scission, nagbawas tayo ng isang block. Dito sa cross-linking, nagdadagdag tayo ng block or blocks. Kaya mas nagiging malapot siya. No? And we have these point lesions. So radiation interaction with macromolecules also result in disruption of a single bond, producing point lesions. So point lesions are not detectable, okay, but they can cause minor modification of the molecule, which in turn cause to malfunction within the cell. So like for example, may na-detect tayo ditong point lesion. Kaya this in a way, halimbawa isang cell or isang macromolecule ng cell, masasabi natin, nagkakaroon na siya ng parang hubag-hubag, di ba? Pag lesion nga ni hubag man to. Ayun. So this explains why there is a possibility of, yun na nga, tinatawag nating cancer. So at low radiation doses, point lesions are considered to be the cellular radiation damage that results in stochastic radiation effects observed at the whole body level. Kasi sabi nga natin, di ba, mga cancers, yan. So late effects of radiation, these are stochastic or late effect, yan, late effect of radiation. So sabi nga, low radiation doses, kaya it's not good for us that, halimbawa maging radtech ka, you keep on parang nag-volunteer ka na nga. Halimbawa, may na yung pasyente nga bata, okay, tapos di rin na-cooperate ang baby, tapos nag-handle ang baby is only the mother. Sige nga. Sige, mapa-expose na laak. kaptang bata, it's not good kasi ano siya, we are to emphasize what we call the stochastic effect of radiation, di ba? Long-term effect of radiation. Although it's in chronic exposure, yung sige-sige. So this can, in a way, may tinatawag tayong point lesions, di ba? At low radiation doses, point lesions are considered to be the cellular radiation damage. Okay? That results in a stochastic effects observed at the whole body level. So that's why you, as the future radiologic technologists, we would like to emphasize that we must observe then radiation protection from or for ourselves as radtech. Diri maupay nga sige kitahin ka-expose with the radiation. So, laboratory experiments have shown that these types of radiation effects on macromolecules are reversible. Okay. So, sabi nga natin, yan. So, may nafa-follow tayong 4 R's in radiobiology, di ba? So, radiation, response, repopulation, and recovery. That's why, so, as a conclusion ng mga scientists, di ba? So, yung main chain scission, yung cross-linking, yung point lesion, pwedeng bumalik sa original na macromolecule. Because, again, this is of intracellular repair and recovery. That's why our human body, is so complex that we can still recover at any amount, okay, amount of radiation. Okay, let's talk about these modern or these molecular synthesis. So, halimbawa, nagsisynthesis ka nga ni, di ba? Photosynthesis, yan, di ba? So, we use light para mag-grow and plant, di ba? Yan. So, para so here, for macromolecular synthesis, parang it's a way magproproduce tayo ng bagong macromolecule, tapos kailangan ipamature natin yung bagong molecule na yan. No? So modern molecular biology has developed a generalized scheme for the function of normal human cell. So that's why yung mga pinag-aaralan natin ngayon is in-depth ng biology, So in a way, nagiging review nito yung mga former concepts when you were in high school, especially if you graduated from STEM, di ba? The science, technology, engineering, and mathematics. But anyway, I hope you can have a little review on this one kasi our book is so limited, so we are expecting that you have foundation on this one, so we might not as well include these concepts here. So molecular nutrients are brought to the cell and are diffused through the cell membrane, where they are broken down, catabolism, diba? If we can have a review, if we can still remember that metabolism, ayun, ang pangit ng pagkasulat pag naka-mouse. Okay, metabolism is divided or composed of 2 segments. We have catabolism, which is they are broken, or the big molecules are broken into smaller molecules with accompanying release of energy. Like for this example, catabolism, breakage or breakdown of large molecule. So if this is a long chain macromolecule, so kailangan buwagin siya, but para mabuwag siya, kailangan meron tayong energy. Magre-release siya ng energy, di ba? Like for example, yung yung carbohydrates, di ba? Para ma-broken down siya, kailangan para ma-digest siya, intracellular digestion. So, magre-release siya dapat ng energy, Kaya, 'di ba, sabi nga ng teacher natin when we were still in biology class, kailangan gumagalaw tayo, galaw-galaw din pag may time para hindi tayo ma-stroke. Kasi if this energy are not used up, nagtataba tayo. Diba? Wala ba dito si Ortego para makasabihin ano eh, makasabihin may bias ako. Ayan, so halimbawa yon, kailangan mag-exercise kita kaya nga ni may na bago nga gym at UEP. Kay para aton mga ginкакаon, so nada-digest, nag-undergo siya ng catabolism. Okay, so that okay, yung energy kailangan gamitin natin. So the other segment, the anabolism, so synthesis of larger molecules. So kung makita natin from smaller to bigger. So this energy is used in several pathways, but most of the more important ways is that they are used in construction or synthesis of macromolecules from smaller molecules. That's what we call the anabolism. So yung energy na nagagamit, na-release ng catabolism, pwede siyang magamit for anabolism. So here, na-broken down yung bigger molecules to smaller molecules. Dito naman, separate molecule, tapos pwede natin gawing mas malaking molecule. So yung energy dito, pwede natin gamitin dito. Okay? So the synthesis of protein and nucleic acids is critical to the survival of the cell and to its reproduction. So let's try to watch this video para mas maintindihan natin at para may break din naman ako kahit pa paano. Okay, so keep lang, keep. And then I'm going to accept the this is one of the perks of having classes online kasi you can inject mga ayun na nga, mga materials available online para mas magamit natin for our discussion. Kala ko may papasok, wala pala. Okay, so for a while, let's see pa yung nandito.
So what is ClickUp? ClickUp is an all-in-one productivity software where you can manage everything and anything related to...
Hello friends, today we will discuss the concept of metabolism. So whatever we eat, whether it contains proteins, carbohydrates or lipids, all these are digested and converted into their simpler form in our body. Proteins are converted into the amino acids. Complex carbohydrates like starch, they are converted into simpler sugars, and lipids are converted into fatty acids. Now during this process, the energy is released in the form of ATP, and other energy rich molecules like NADH2 or FADH2, they are also formed and which can be used for different purposes. Some waste material like nitrogen compounds, CO2, phosphates and sulfates, they are also produced during this process. Such waste material is excreted outside the body by different ways. Now these simple molecules can be again used to produce different complex and large molecules which can play different role in our body. For example, enzymes, the hormones, the receptors, and even the hair and nails. So as you can see, whatever we eat, it changes from one form to the other. And in Greek language, the word change is called as metabolismos. And from this Greek word, the new term came into the existence in science, the metabolism. So metabolism can be defined as sum of the total of all these biochemical reactions occurring in a cell. And this interconnecting network of reactions are called as metabolic pathways. Metabolism can be classified into two categories: catabolism or catabolic reactions, and anabolism or anabolic reactions. Catabolic reactions are those in which larger and complex molecules are degraded into smaller and simple molecules. And during these reactions, energy is released in the form of ATP. For example, conversion of proteins into amino acids and conversion of lipid into fatty acids. A classic example of catabolism is the degradation of glucose molecule into pyruvate. Glucose is hexose sugar, that is, it is made up of 6 carbon, and pyruvate is made up of 3 carbon, that is triose. This degradation occur in 10 stepwise reactions, which is known as glycolysis. And during this conversion, two ATP molecules are produced. Anabolism is exact reverse of catabolism. In anabolism or anabolic reactions, small and simpler molecules, they are converted into large and complex molecule. And during these kind of reactions, energy is consumed. In catabolism, energy is released, and in anabolism, energy is consumed. For example, conversion of amino acid to keratin protein, which is present in the hair and nail. Another example is synthesis of glycogen or starch from glucose molecules. So this was all about the basic concept of metabolism. Thank you very much for watching this video, and if you like this video, please like and subscribe my channel.
So ayun ha, na na-remind kita guys, whatsoever that you intake, kailangan i-broken down naton in ginkukonsume naton lawas. So after diri la kay kutob la kita kaon, kailangan nag-exercise ngayon kita para may the energy nagagamiton for ayun, mga kinakaon naton, they are being catabolized, no? And ito nga mga nutrients na maka-absorb for from the catabolism, himon lot for bigger molecule for our growth, okay, for our living. Yan. So an ATP, the energy, that's what we call adenosine triphosphate, ang ginagamit for cellular respiration. Take note, diba, like what we said the last time for human biology, cellular respiration na occur, diba, powerhouse of the cell in the mitochondrion. So all of these things, the mga cellular digestion, catabolism, anabolism, happen in the mitochondrion, the powerhouse or the workhorses of a cell. So yan. So kailangan pagkaon, exercise, kasi ating ginakaon broken down into smaller, molecules, and then these molecules will be utilized again for gagamitin doon for growth. Parang bata, kailangan natubo. That's the part of human life. Okay? So, ayun, I hope we really had a gist on that and we understood this concept para if ever we have a class next week, tikmarong kung holiday, so we can have a chapter examination on this one. And alam niyo naman, diba, sabi ko nga, yung PowerPoint is only a guide, most of which cannot be, di rin na natin mabubutang all of the concepts here, especially kung mga ganun, these concepts were taken when you were in high school. Okay, so we have the molecular synthesis. Okay, so sabi nga natin kanina, diba, parang photosynthesis, diba, ginagamit natin ang light para mag-grow, diba? Magproproduce kita ng mga bagong macromolecule and kailangan ipamature natin yung mga macromolecule. Kaya lang may synthesis para for our growth and development. And so here, proteins are manufactured by translation. So duhamat naka-highlight, color blue. We have translation of the genetic code. from transfer RNA, which had been transferred from messenger RNA. So the information carried by the mRNA was in turn transcribed from DNA. So here, kanina naton molecular synthesis naging discuss, generally siya general for the human body or general molecule. So we need to be specific here in the discussion. Punta tayo kay protein. So yun, yan. Punta tayo kay protein. So kailangan mag-synthesize yung protein. E di ba protein damot iya damot iya gin ako para nga trabaho hala aton lawas, di ba? So si protein na act as enzyme, it acts as hormone, it acts as antibody. So kung kulang ang aton protein, dire ma-function ang aton iba-ibang hormones, di ba? So dire siya ma-function ng iba-ibang in form of enzymes and kulang ang aton defense mechanism in our human body. So para daw makahimon bago nga protein, so may duha nga process, translation and transcription. Parang nagano lang, parang naghihimon parang nagkakalat-kalat chismis ano. So kung halimbawa, anong bagaw ni ano, anong gin bagaw ni ang taga section B, ay bagaw ni ko ano, kita-transcribe mo tapos ito na-translate na into bagong information. So meaning si DNA an may hawak ng lahat ng ano ng information para kailangan na ni cell mag-grow. Kailangan magdamo kita DNA. So DNA hin protein. So uncode or uncommand matikang kan DNA. So kailangan muna may i-transcribe na message. Ma-accomplish ni because of mRNA. So from there, from the mRNA, kailangan i-transfer to other cells. Especially that we know that proteins are being synthesized in the ribosome. Diba? Kanina, ang ibang macromolecule, ginsynthesize siya dito sa mitochondrion. So here, proteins are synthesized in the ribosomes. So duha nga process, ano. So translation and transcription. Okay, sige. Panoorin muna natin ang video na to para mas maintindihan natin. Wala, sorry. Why ko ngayon na-share na itong screen kanina? So, ito yung sinasabi. Sorry, ha. Sorry, hindi ko na-share yung screen ko. Ito pala yun. Ito yung sinasabi ko kanina. Nag-unala ako. Nag-una-unala ako. Ayun. So, ito yung sinasabi ko kanina na macromolecule synthesis. So proteins are manufactured by translation of the genetic code from transfer RNA, which had been transferred from messenger RNA. So the information carried by the mRNA was transcribed from the DNA. So if you look into the picture of a cell, alam naman natin yung DNA, di ba, nandun sa nucleus. Yan. So DNA nasa nucleus. Siya. So we can see this one as the nucleus of the cell. And then, this is the bulk of the cell, the cytoplasm. So si DNA, magkakamand na siya na, ay, we need to have additional proteins sa human body. Kasi nga, daming function ni protein eh. Hormones, enzymes, antibodies. So yung command, magmumula dito sa nucleus. Nandito si DNA. Siya yung mastermind. So ma-accomplish niya yan kasi may mga mRNA tayo, mga messenger, at saka may mga transfer RNA. So ito, this is the ribosome. This is the part of the cell where protein are being synthesized. Walang iba. Wala ng ibang part ng cell. Sa hindi sa lysosome kasi ibang function ni lysosome, ibang function ni endoplasmic reticulum, iba yung function ni mitochondrion, doon lang sa ribosome. How this can accomplish? Sige, panoorin natin yung video na to.
user experience than not having a live chat at all. But there's a better way. Use 8
Diretso
base.
ah.
With 8 base, you can teach an AI chatbot.
Skip.
After learning about DNA, have you ever wondered how can the DNA actually result in a trait? Let's take an example, like eye color. Yes, your DNA has the genetic information that codes for the color of your eyes. Your eye color is based on a pigment that is inside the eyes, but in order to have that pigment, you have genes, which are portions of DNA that can code for proteins which help make that pigment. So what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something, so protein synthesis means to make protein. And you may wonder, what's the big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. You've got to make proteins. It's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA. Well, a few exceptions. And that DNA is in the nucleus. Some DNA is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our gene regulation video, but we're going to talk about genes that are coding for active proteins. So how are we going to get the information from these genes out of the nucleus so that the cell can start producing the proteins that it needs to make? Well, let us introduce you to the amazing work of RNA. We have a video comparing and contrasting RNA and DNA. We'll be brief here in saying that RNA is a nucleic acid, like DNA, but it has a few differences. Its role in protein synthesis also is huge. Before we get into the process, please note our typical disclaimer. We tend to simplify topics, but as always, we hope that you have the desire to explore this complex, amazing process later on, to learn all about the extra information that we don't have the ability to include in this short video. In protein synthesis, we can look at two major steps. One is transcription and the other is translation. Transcription has a C in it, and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. The transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You could see on a codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. The tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished. So the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA, so the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA. Protein folding and modification may occur, and the protein may need to be transported. This can all vary based on the protein structure and function. Another fascinating topic for another Amoeba Sisters video. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So ayun, so nakita natin ang Amoeba Sisters. So kahit pa paano, di ba, we established concepts related to protein synthesis. So yan na, in a way, magkakaroon na kita, Tagalog ko kayari. So magkakaroon na kita idea, di ba, we have 22 amino acids. Yung 22 pala yun, may mga names, di ba, mga proline, methionine, di ba, mga valine, leucine. So we can have names on that one. But anyway, so kahit pa paano, nare-review kita and how proteins are being synthesized. So again, sabi nga ng Amoeba Sisters, why? Bakit big deal yung protein? Because take note, on our lecture on human biology, that protein protein has so many functions in the human body. So kailangan natin ng protein sa ating katawan. No? Okay, so let's continue. Mo talaga maupay ko nag-online class. We have the perks of having online class kasi we are having a film showing at the same time. And later, pag masend ko na niyo video, mapenta ko natin yung mga nawungkay para makikita taliwat talagang full and out on discussions. Madali yung maka-review. Hopefully, yes. Okay, so let's continue with our discussion. I hope okay pa ka mo nga da. Isa na po board na ka mo. Charit. Pag timbre la ka mo, I guess, I don't know if Dermalin can also handle a class because may emergency din siya ngayon. Rest kami. But anyway, so let's continue your supposedly your schedule is 9 a.m. pa naman, di ba? So I still have time. Okay, so let's continue on this one. So radiation damage to any of these macromolecules may result in cell death or late stochastic effect. Okay, so but good thing is kasi bisan kita sige ka-expose, radiation damage to any of these macromolecules will result in cell death or late stochastic effect. That's why proteins are continuously sensitized throughout the cell cycle and occur in much more abundance than the nucleic acids. So, bisan sige man kita ka-expose to radiation, bisan sige kita, di ba, di man lang radiation X-ray. So there are many forms of radiation. And proteins are being continuously synthesized. Kaya talagang talagang hindi tayo ganun kamarupok. Lumalaban tayo, of course. So, mas madamo ang synthesize nga protein compared with amino acids. Okay, sorry. The nucleic acids. So take note that this chain of mga bilog-bilog na yan, so mga amino acids are building blocks or building blocks of proteins. So furthermore, multiple copies of specific protein molecules are always present in the cell. So consequently, proteins are less radiosensitive than nucleic acids. So the nucleic acids kasi are, diba, some of the nucleic acids carries genetic code for the human body. Okay, so that here we can establish the contrasting idea. So if we differentiate protein and nucleic acids into its radiosensitivity, so masasabi natin radiosensitive pa din yung mga nucleic acids, especially yung nagdadala ng mga nucleic acids, diba, sa DNA. Yan. So we cannot explain here how lipids, how carbohydrates are being synthesized kasi yung function naman nila is more of yung lipids, alam naman natin, these are thermal fashion. Yung carbohydrates, this is energy, for fuel the body. But here, dalawa yung pinag-uusapan natin dito, mga major macromolecule na kailangan i-synthesize. Okay, so we are already done with proteins. Yay! So now let's go to the amino acids. So similarly, multiple copies of both types of RNA molecules are present in the cell. Although they are less abundant in protein than protein molecules, on the other hand, the DNA molecule with its unique assembly of bases is not so abundant. So di ba may mga base tayo, di ba, mga adenine, thymine, cytosine, guanine, yan. And DNA is the most radio-sensitive molecule. So here, parang hindi naman to ganun ka-relevant yung picture. This is crystallogram. Okay, so it's just that pinapakita lang natin yung clearer picture ng hopefully makita yung picture ng DNA. Okay, so alam naman natin that DNA is in the nucleus of an atom. But based sa biology, not all atom, bakit atom? Cell. So the nucleus of a cell. So based on the biology, hindi lahat ng cell are may nucleus. Diba may tinatawag tayong eukaryotes or eukaryotic cells. Yan, sila yung may ano, sila yung may mga nucleus. Yung yung walang nucleus, tinatawag nating prokaryotes, prokaryotic cell. Okay, so how these DNA are being synthesized? Sorry, yeah, how is DNA being synthesized? So DNA is synthesized somewhat different from proteins. So during G1, 'di ba, alam naman natin yung yung cycle ng mitosis, 'di ba? So we have the S tapos G1. So during G1 portion of the interphase, the deoxyribose, the phosphate, and the base molecules accumulate in the nucleus. So here, parang yan. Parang nagiging evident or nagiging visible yung mga bases natin during G1 or the pre-synthesis phase. So these molecules combine to form a single large molecule that during the synthesis portion of the interphase, this is attached to an existing single chain of DNA. Medyo complicated siya, no? May G1, yan. So, parang ihihiwalay natin yung mga bases. Yan. Kasi di ba? So, during G1, the deoxyribose, the phosphate, and base molecules accumulate in the nucleus. And these molecules combine to form a single large molecule during the S portion is attached to an existing single chain of DNA. Okay, this is in the familiar double helix form. So half as much DNA is present in G1 as in G2, post-synthesis. So as the cell moves into the S phase, the ladder begins to open up. Okay, during synthesis, now open yung ladder, okay, of each rung like a zipper. So parang di ba, double helix. So yung 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 rung now open up. Charan! And now the DNA consists of only a single chain and no pairing of bases. So na-open na, yan, open fec na siya. One man like to expression. Yan. So here, the DNA consists of only a single chain and no pairing of bases. Kita niyo, A, T, C, yan. Single. Okay, so yes, so ganun, giputol kasi dito ni Busyong. So to complement this this discussion, so let's watch this video from YouTube. That's you. So here you please take note of the different proteins, the helicase, the ligase, the polymerase, pa yun? Yeah, the primase. So kailangan tandaan natin yan, yung mga leading strand, yung mga lagging strand, ayun, yung mga Okazaki fragments, everything that the video will talk or will say to you. Oh.
DNA. We talk about it so much. It's the ultimate director for cells, and it codes for your traits.
Dali lang.
DNA. We talk about it so much. It's the ultimate director for cells, and it codes for your traits. With a molecule that has a function like that, it makes sense that when you make another cell, like in cell division, you would also need to get more DNA into the new daughter cell. And that introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where? Well, if it's in a eukaryotic cell, it occurs in the nucleus. However, remember, not all cells have a nucleus, such as prokaryotic cells. They don't have a nucleus. Still, both prokaryotic and eukaryotic cells do DNA replication, but there's some differences between the two that this clip doesn't go into. Next, when? When does this happen? Well, a cell is going to need to do this before it divides so that the new daughter cell can also get a copy of DNA. To get specific, in a eukaryotic cell, that's going to be before mitosis or meiosis in a time known as interphase. I think DNA replication would actually make a great video game. Still waiting for that to be invented. I'm going to introduce the key players in DNA replication so that you can get some background information. Now remember, these are just some major key players. There's a lot to this process. Many of the key players are enzymes. In biology, when you see something end in ASE, you might want to check, as it's very possible that it's an enzyme. Enzymes have the ability to speed up reactions and build up or break down the items that they act upon. So here we go with the key players. Helicase, the unzipping enzyme. If you recall that DNA has two strands, you could think of helicase unzipping the two strands of DNA. Helicase doesn't have a hard time doing that. When unzipping, it breaks through the hydrogen bonds that hold the DNA bases together. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer it makes? The primer is actually made of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you would need that later on. Now, don't feel overwhelmed. We'll go through the basics of this sequence in order. But remember, like all of our videos, we tend to give the big picture. There are always more details and exceptions to every biological process that we can't include in such a short video. DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. At the origin, helicase, the unzipping enzyme, comes in and unwinds the DNA. Here's the thing though, you don't want these strands to come back together. So SSB proteins, which stands for single-stranded binding proteins, bind to the DNA strands to keep them separated. And topoisomerase, I always have to slow down when I say that enzyme's name, keeps the DNA from supercoiling. Supercoiling might sound super, and it can be when you're trying to compact the DNA, but it's something that needs to be controlled during DNA replication. Supercoiling can involve an overwinding of the DNA, and you need the DNA strands to be separated for the next steps. Primase comes in and makes RNA primers on both strands. This is really important because otherwise DNA polymerase won't know where to start. In comes DNA polymerase. Okay, before we go on, remember how we said DNA has two strands? They're not identical, they complement each other. In our video that covers DNA structure, we talk about how the bases pair together with hydrogen bonds. The base adenine goes with the base thymine, and the base guanine goes with the base cytosine. These strands are also anti-parallel, so they don't go in the same direction. What do we mean by direction? Well, with DNA, we don't say north or south. We say DNA either goes 5' to 3' or 3' to 5'. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of this ring structure. Now, you do the same thing for the other side, but keep in mind DNA strands are anti-parallel to each other. So let's count these, again clockwise after the oxygen. 1', 2', 3', 4', 5'. And the 5' is out of this ring. This strand on the left runs 5' to 3', and the strand here on the right runs 3' to 5'. We'll explain why all that matters in a moment. So let's take that knowledge there and look at DNA replication here. In this image, I labeled the top original strand 3' to 5'. I labeled this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helicase. In this example, it will keep unwinding in this direction. Primase places primers. DNA polymerase is building the new strands. Now, the thing about DNA polymerase is when it's building a new strand, it can only build the new strand in the 5' to 3' direction, meaning it adds new bases to the 3' end on the new strand. See how it's being built in the 5' to 3' direction? This one is called the leading strand. But take a look down here. So DNA polymerase once again is building a new strand in the 5' to 3' direction. But there's a bit of a problem here. See, as DNA unwinds, because DNA polymerase can only build the new strand in the 5' to 3' direction, it has to keep racing up here next to where this unwinding is happening. You can see why then this new strand is known as the lagging strand. On this lagging strand, primers have to keep being placed in order for DNA polymerase to build. These fragments that result are known as Okazaki fragments. Primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, the gluing enzyme, as I like to nickname it, has to take care of the gaps between Okazaki fragments, sealing them together. At the end of replicating, you have two identical double helix DNA molecules from your one original double helix DNA molecule. We call it semi-conservative because the two copies each contain one old original strand and one newly made one. One last thing. Surely you've had to proofread your work before to catch errors. In this process, you don't want DNA polymerase to make errors. If it matches the wrong DNA bases, then you could have an incorrectly coded gene, which could ultimately end up in an incorrect protein or no protein. DNA polymerase is awesome. It has proofreading ability, meaning it rarely makes a mistake, which is a good thing. So remember how we said there is far more detail to this process to explore? The detailed understanding of DNA replication has led to some life-saving medical treatments that can stop DNA replication in harmful cells, including pathogenic bacteria or human cancer cells. We encourage you to explore beyond the basics. Check out the further reading suggestions and the video details to explore more. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So yun, nakita natin ano, na ginhatag ng mga ideas. Yeah, right ideas from the Amoeba Sisters. Thank you so much. Kaya sabi ko nga, this is the perks of having your classes on online kay nagagamitan gihapon ni nato at mga references or mga videos from, like for example from YouTube. And anyway, after this discussion, our videos will also be posted on YouTube. Yan. So, di ba na kung help? But anyway, so I hope mas naintindihan natin kasi yung nabasa natin kanina sa busyong, medyo cut siya, di ba? Nautod siya. Mas damo sa sa G1, G2, mas damo sa G2 kaysa sa G1. Kasi nga may mga bago na, di ba? May mga semi-conservative na mga strands. So may usang arang, that's from the old one. May bagong arang na parang na para sa mga newly constructed one. So as I've said, some of the concepts here are not included, of course, kay limited it atong reference. So, but I encourage you and I advise you, of course, kay para sa next chapter examination, damot mala answer natin. Okay, so let's continue with our discussion. Kasi ang dami kasing ang daming molecules sa ating katawan, di ba? 5 major molecules. Dalawa pa lang yung napag-uusapan natin. But anyway, hindi natin pag-uusapan na yung dalawa, yung lipids at saka yung carbohydrates. Saved by the bell. Kasi when you talk about radiation, so radiation interaction with matter, di ba? Normally, yung lipids at saka yung proteins, yung lipids at yung carbohydrates, they are yung mga superficial layers of the skin. especially lipids. So they are considered ano siya, radio-resistant. So doon tayo sa ibang mga macromolecule, doon tayo sa ibang molecules. Okay, so let's continue. I hope ada pa kamo dida and willing pa mamati akon until 9 a.m. 9 a.m. but I don't know. Okay, so okay, let's continue again. So we're done with the two macromolecules, the proteins and nucleic acids. So now let's talk about the radiation effects on DNA. So as we said, DNA is the most important molecule in the human body because it contains the genetic information for each cell. Okay, genetic information. So, kung halimbawa mo mga magiging future, future na anak, future descendants, si DNA ang may hawak niyan, no? Each cell has a nucleus that contains DNA complexed with other molecules in the form of chromosomes. Yan, kasi 'di ba, take note, for each of us has 23 pairs of chromosomes. 23XX or 23XY. So chromosomes therefore control the growth and development of the cell, and this in turn determine the characteristics of an individual. Kasi nga, nandyan ang genetic code sa chromosomes, especially nasa nucleic acids or kay DNA. Okay, so if radiation damage to the DNA is severe enough, visible chromosome aberrations may be detected. Take note na lang of this term. I am not sure if this will be this will be covered on probably on late effect of radiation, chromosome aberration, but naghatag na tayong advanced na term, chromosome aberration. So again, if radiation damage to the DNA is severe enough, so visible chromosome aberration may be detected. So the DNA molecule can also be damaged without the production of chromosome aberration. So dalawa yung pwedeng mangyari sa radiation, ano? Pwedeng chromosomal aberration or pwedeng DNA itself yung maging target. So although such damage is reversible, so it can lead to cell death. So kung mga mga chromosome aberration, pwedeng balik. Pero pag halimbawa cell death na yan, may babalik kasi patay na yung cell, di ba? Anyway, our human body is so complex. Ang dami naman tayong cells in the human body can be replaced. So if enough cells of the same type respond similarly, then a particular tissue or organ can be destroyed. Yun yung magiging catch natin dyan. But that describes the cause of deterministic effect. Deterministic, again, this is the early effect of radiation. So, paano? So, either for chromosomal aberration or for DNA of the same cell or the same particular tissue or organ. And damage to the DNA can also result in abnormal metabolic activity, uncontrolled rapid proliferation. Di ba, proliferation, this is the process of the cell division or of a cell multiplying in number. And this is the principal characteristic of a radiation-induced malignant disease. Malignant or cancer. Kasi nga, na siya, hindi na natin nakokontrol ang kanyang pagdami ng cell. Di ba? So, nagkakaroon na ng swelling, di ba? Swelling or lesion, tawag nating tumor. So, if the tumor is malignant, then that is a cancer. So, that's why it describes the cause of stochastic effect. Stochastic, or this is the late effect of radiation. So, ulitin ko lang yung chromosomal aberration, this is not yet covered in molecular radiobiology. This is covered in either, deterministic effect of radiation pala. Deterministic, so si chromosomal aberration. So, the early effect of radiation na discussion. Okay, so if if the damage to the DNA occurs within a germ cell, diba? Germ cell, germinal cell, tawag natin, these are for reproduction, diba? Reproductive cells. Then it is possible that the response to the radiation exposure will not be observed until the following generation or even later. So this describes the cause of genetic effect. So halimbawa, ikaw na parent, okay, you were exposed to radiation when you were still younger. Yung effect pala pwedeng makita sa next generation mo. Like halimbawang si girl na loyal kay mister, tapos nagkaanak ng kamukha ni The Hook, yung movie kanina. So sasabihin ng asawa, hindi ko anak yan, sinong ama niyan? Tapos masaring babae nga. Wala naman akong ibang lalaki na minahal, ikaw lang. O di ba, may tayo sa drama. So, it's because of the genetic effect of radiation na pwedeng nakuha pala ni Mrs. when she was still younger. So yan, this explains the radiation effects on DNA. Kasi nga si DNA ang may hawak ng genetic code. So at some point in her life, na-disrupt because of radiation. So pwedeng makita yung effect on the next generation. Yes. So the chromosome contains miles of DNA. Therefore, when a visible aberration does appear, so it signifies a considerable amount of radiation damage. So unobserved damage to the DNA can also produce responses at cellular and whole body levels. And these types of damage can occur at the DNA, and these are described as follows. 1. This can be a main chain scission with only one side rail severed. So, halimbawa, this is the DNA double helix, di ba? Double helix, shape. So, sabi dito, pwedeng isang side rail lang ang maapektuhan. Pwede naman main chain scission with both side rail severed. Main chain scission and subsequent cross-linking, yung natanggal ditong chain, makakabit sa ibang DNA. Rung breakage causing separation of bases and a change or loss of a base. Ito yung matindi, change or loss of a base. So although each of these effects results in structural change in the DNA molecule, so they are all reversible. Yan, they are all reversible. So yung iba, yan. In some of these types of damage, the sequence of bases can be altered. Therefore, the triplet codons may not be or may not remain intact. Codons, di ba, yung na kanina, di ba, mga CCU, UEP, UEA, DKEU, yan. So, mga codons yan, mga codes. So, may not remain intact. So, therefore, magkakaroon ng chromosomal aberration or variation ng chromosomes. So, this represents a genetic mutation at the molecular level. So, pwede nga, halimbawa, if genetic code, if it is a germ cell, yun, pwede nang magkaroon ng mutant, yung magiging anak mo. Magkakaanak ka na ng isang Jean Grey, isang Wolverine, yan. Kasi, na-alter, yan. May chromosomal aberration. Yan. So, anyway, itong PowerPoint, nadi-discuss lang siya ng Down syndrome. Yan. Okay, what else? The fifth type of damage, the damage or loss of a base, this is masyadong matindi. Also destroys the triplet code and may not be reversible. Triplet code, di ba? Halimbawa, UEP. Ayan, natanggal ang P. So, di na siya complete yung codon. So, magkakaroon na tayo ng abnormality. This type of damage is a molecular lesion of the DNA. These molecular lesions are called point mutations, and they can be of minor or major importance to the cell, yung inala. So, pwede dito magkaroon ng abnormality. Yan, pwede na point lesion na tinawag. Pwedeng maging point na siya ng cancer. Diba? So one critical consequence of point mutation is the transfer of incorrect genetic code to one of the two daughter cells. Yun, pwede nang mo anak, pwede na may cleft palate, wag naman, yan, diba? Kasi mali na ang genetic code niya na transfer doon sa next generation. So the three principal observable effects that may result from irradiation of DNA, ayun, cell death. Buti na lang maraming cells sa ating body, di ba? So, pwede nating masabing, okay lang, naging resistant naman tayo. But here, malignant cell, abnormal proliferation na kasi yung cells, kaya nagiging ano na siya, nagiging mabilisan na yung pagdami ng cells, so nagiging malignant, no? And genetic damage. Gaya ng sinasabi natin kanina, na halimbawa nagkaanak ka na ng Wolverine, ng Jean Grey, sino pa yun, ni Cyclops. So the latter two effects at the molecular level results in stochastic responses and conform to the linear non-threshold dose-response relationship. Linear non-threshold dose-response relationship, itong dalawang ito. So, pang matagalan. No? Okay, so that's it. The molecular radiobiology, particularly on nucleic acids and the proteins. Okay, sabi natin kanina, yung water, iba kasi yung kanyang interaction, no, sa radiation. So we have a specific topic which is radiolysis of water. But we don't need to to explain how water are being synthesized, kaya talagang we are advised naman, 'di ba, to take, 'di ba, 8 to 10 glasses of water a day, lalo na ngayon, this is summer, we need to be hydrated. So, normally yung water ini-intake talaga natin, hindi siya nare-reproduce sa human body. But anyway, so because of the said substance that the human body is aqueous solution, it contains 80% of water molecules. So irradiation of water represents the principal radiation interaction in the body. Okay, so meaning yung katawan natin talagang tubig, 'di ba? Water phantom. Maliban lang sa iba dyan na nagsasabing plastic sila, hindi kami dyan, hindi kami niyan. Tawan ko lang, wow, walang ganon. So 80% water. Kahit sabihin lang plastic kayo, basta alam natin you are composed of 80% water. And when water is irradiated, it dissociates into other molecular products. No, from H2O, diba, this is the chemical formula for water. So pwedeng mahiwalay, no? Pwedeng sirain ng radiation. Wow. This action is called radiolysis of water. So class, pag makita niyo yung radiolysis, okay, wala na kayong ibang iisipin yan. So ito na yung radiation interaction sa water inside the human body, no? Radiolysis. So when an atom of water is irradiated, it is ionized and dissociates into two ions. So that's what we call an ion pair. So ito yung chemical formula ng water. This is a radiation. So after ma-expose sa radiation, sabi nga, dalawa yung maproproduce na ion. Kaya ion pair. Isang positive at isang negative. What else? After this initial ionization, a number of reactions can happen. First, the ion pair may rejoin into a stable water molecule. So in this case, no damage occurs. So again, pwedeng isang molecule, especially the positive, mag-join sa water, wala, okay lang. Wala namang mangyayari. So second, if these ions do not rejoin, it is possible for the negative ion, the second electron, to attach to another water molecule through which reaction produce a third type of ion. So if the negative ion joins again a stable a water molecule, so pwedeng ma-produce na ng another ion, negative ion. So the HOH positive and the HOH negative are relatively unstable and can dissociate into smaller molecules as follows. So pwedeng mahiwalay yung HOH positive. So, isang positive ion and then the OH. So, bilangin natin, kailangan sa chemistry kasi kailangan kung ilan yung molecules dito sa left side, ganun din naman yung, atoms. Kung ilan yung atoms dito sa left side, ganun din ang bilang natin ng number of atoms on the right side. Kung may dalawang atoms ng hydrogen dito sa left side, kailangan may dalawa siyang atoms dito sa sa right side. Yan. Kailangan balance. So the final result of radiolysis is the formation of an ion pair, isang positive and isang negative. Yan. So isang positive at isang negative. The H positive and the OH negative and two free radicals. Nakita nyo may mga asterisk na sign. So these are called the free radicals. So again, ito yung pinaka-main na mangyayari after radiolysis. Dalawang ion, OH positive and OH negative, and dalawang free radicals. So these ions can recombine, therefore no biologic damage occur. Okay lang kung ions lang, ano? But ibang story pag free radicals. So a free radical is an uncharged molecule that contains a single unpaired electron in the outer shell. Okay, so this is the definition of free radical. So free radicals are another story. They are highly reactive. Free radicals are unstable and therefore exist in a lifetime of less than 1 millisecond. And during that time, however, they are capable of diffusion through the cell and interaction at distant site. So pwede palang mangyari, halimbawa si ba di mak nangingilalagot? Sige daw, i-exit ko muna. Pinaparitak attendance. So, for example, he Fraga ginaway ni Disolok. So, sabi 'di ba sabi sa ating notes, 'di ba, it can produce interaction at distant site. So, halimbawa, ganito yung ganito yung nangyayari pag mga free radicals. So, for example, si Fraga at saka si Disolok nag-away. Tapos, kay magbeshiwap si Fraga at saka si Iskilyo. So, pwedeng si Iskilyo yung maging pinaka-affected sa lahat. O bisa ni Fraga ni Disolok ang nag-away, pwedeng si Iskilyo ang magpro-post sa Facebook. Shoot ka girl. O ganon. So, pwedeng maging affected ng bonggang-bongga. Kaya there is now the free radical na hinabok because of Fraga, So that is pwedeng maging simple analogy with the atom or the human body. So pwede tayong halimbawa yung na-irradiate is, let's say, lungs, radiation therapy ng lungs, tapos nagkaroon ng liver cancer. Pwedeng mangyari because sabi nga natin, the human body is aqueous in solution. Or the other way around. Meron tayong pasyente, halimbawa liver cancer, liver irradiation, tapos nagkaroon siya ng, let's say, ovarian cancer. Yan, pwede yan kasi there is now what we call the radiolysis, distant effect ng radiation. So again, free radicals are capable of diffusion through the cell and integration at distant site. Free radicals contain excess energy that can be transferred to other molecules to disrupt bonds and produce point lesions at some distance from the ionizing event. Now, again, dalawa ng laman yung free radicals natin, H and OH free radicals. These are not the only, kanina kasi sabi dalawa, but again, they are not the only free radicals that are produced during radiolysis of water. So, aside from the H and the OH, pwede pa lang, okay, the OH free radical can join with similar molecule to form hydrogen peroxide. So, dalawang OH, so dalawang atom ng hydrogen, H2, dalawang atom ng oxygen, O2. So, H2O2. Remember that hydrogen peroxide, tama ba ni agwa oxinada, is poisonous to the cell and therefore acts as a toxic agent. Yan. So, that's the jar. And here, the H free radical can interact with molecular oxygen to form the hydroperoxyl radical as follow. So, kasi na-exist na to sa human body, di ba? We take oxygen in, carbon dioxide out. So, pwedeng mag-combine yung free radical at saka yung oxygen. So, HO2. So the hydroperoxyl radical along with hydrogen peroxide is considered to be the principal damaging product after the radiolysis of water. So this explains kung bakit meron tayong mga indirect at direct effect ng radiation. So hydrogen peroxide can also be formed by the interaction of two hydroperoxyl radicals as follows. So dalawang HO2 magko-combine. So dalawang atom ng hydrogen, check. So ito apat na to eh. Dalawa dito and dalawa dito. Yan. So 2 plus 2. So, kailangan balance. Apat din. So, yun. So, that's the byproduct of the 2 hydrogen peroxide. So, some organic molecules symbolized as RH in the red horse, but these are organic molecules, can become reactive free radicals as follows. So, free radicals plus radiation. So, pwede siyang mag-produce ng reactive free radical and pwedeng ma-dissociate into two free radicals. So, when oxygen is present, yet another species of free radical is possible. And, okay, so we have free radical plus oxygen. So, maraming mga pwedeng mangyari in radiolysis. Free radicals are energetic molecules because of their unique structure. So this excess energy can be transferred to the DNA and this can result in band breaks. Yeah, that's the worst thing about radiolysis. Okay. So yan, no? So yung mga magiging effect pala ng, but take note again, this can be hindi naman siya ganun ka, mas alam karadlukan ngayon magpa-X-ray kaya may radiolysis of water. So again, if we can expect that high yung mga magiging effect, kailangan it can be wag tayong papa-expose for mga acute radiation as a radtech, kailangan we need to protect also ourselves. So we have the direct effect and indirect effects of radiation. So when biologic material is irradiated in vivo, or inside the body, the harmful effects of irradiation occur principally because of damage to a particularly sensitive molecule such as the DNA. So evidence for direct effect of radiation comes in vitro experiment, wherein various molecules can be irradiated in solution. So the effect is produced by ionization of the target molecule. So dalawa yung pwedeng maging effect, either direct at pwedeng maging indirect effect. So, if the initial ionizing event occurs on the target molecule, so the effect is radiation is direct. Kung halimbawa, si Disolok at saka si Fraga nag-away, tapos talaga yung magre-react masyado si Fraga, so that's the direct effect of radiation. So, halimbawa, na-irradiate yung lungs, tapos nagka-problema, nagkaroon ng inflammation doon sa lungs, so that can be a direct effect of radiation. Yun. So the other way around, ito yung pinaka on the other hand, if the initial ionizing event occurs on a distant, non-critical to molecule, which then transfer energy of ionization to the target molecule, indirect effect has occurred. So free radicals with this excess energy of reaction are intermediate molecules. They migrate to the target molecule and transfer their energy, which result in damage to the target molecule. So the principal effect of radiation of human is indirect effect. Okay? So yung example natin kanina, like for example, sinabi ko, halimbawa si Disolok, kaya yun talaga din ni Disolok. Ni Fraga man permiti na away. Disolok and Fraga nag-away, tapos si Esquilio ang pinaka-affected. Bakit napasok dito sa picture si Esquilio? Ay mag-beshi, So halimbawa si Esquilio ang pinaka nag-react, nag-propose sa Facebook. So indirect effect. What I'm trying to say is, halimbawa, let's say, may radiation therapy, kaya liver, tapos naapektuhan na niya lungs, so could be that could be an indirect effect ng radiation. Ang may gawa dyan, yung mga free radicals. Okay, so it is not possible to identify whether a given interaction within the target molecule is resulted from direct or indirect effect. However, because the human body consists of 80% water and less than 1% DNA, so it is concluded that essentially all of the effects of irradiation in vivo are from indirect effect. So when oxygen is present, take note, oxygen is a primary cell sensitizer as in living tissue. So the indirect effects are amplified because of additional types of free radicals that are formed. Kasi aside from major cell sensitizer siya, kasi nabanggit kanina, 'di ba? Pwede pa lang may impact si oxygen no dito sa effect ng radiation sa water. So, yun. That ends our discussion for this morning. O, di ba? Natapos gihapon. So, do you have any questions, guys? Are there any questions? Yes, Miss Banan? Hannah?
Excuse me po, sir.
Yes.
Kuan la po, sir. May dala po karo i-clarify po, sir, about san macromolecular synthesis.
Mhm.
Di ba po, sir, after san second video, may ginpaimod mo po, na-mention niyo po about san radiation damage. An akon po
Mm-mm.
question, sir, is that sa kuan po ba, sir, san radiation damage san mga macromolecules, only nagre-result la po ba siya san stochastic effects?
Di ba kanina na-mention man natin nga, depende kung in vitro, kung in vivo. Tapos, an effect natin, we can say deterministic or stochastic effect. Depende sa radiation dose. Depende sa threshold. Depende sa amount of radiation.
Okay po, sir. Thank you, sir.
Okay, so thank you. Are there other questions? So talaga namati talaga yung Miss Baman. I appreciate that. About the others? Parang waray na. Parang feeling ko tuloy ang galing-galing kong teacher kasi waray na napakiana. Okay, sige, let's attendance time. So, yes, Miss Baman. Yes, Hannah? Recognized? You're being recognized? Oh, kala ko may question pa si Hannah. You're raising your hand. Okay, sige, attendance time tayo, guys. Wala pa ngayon.
Ore ko po kapindot ang mic, sorry.
Oh. Yes.
Last na po yun, sir. About po kuan, sir, san indirect effect nga, direct effect po, sir. Nako-consider po ba siya as indirect effect when an ionization po takes place away from the DNA molecules?
Yes. Basta diri siya diri siya talagang target natin na molecule, diri siya naiguan radiation, so pwede siya masabi talaga natin indirect effect.
Okay po, sir. Thank you po, sir.
You're welcome. Yan, sige pa. Hinot na yung pakiana pa. Sige, attendance time tayo. Hala, okay, sige. So as I've said kanina, kailangan naka-open ang camera para makita ta talagang kamutaada. Wow. Diba yun talaga nangaturog na kamo. Maximum naman, maximum. Yan, full screen ko lahat. Ano ko mafu-full screen? Yun. Okay, wow. Mga fresh, sana all. Sana all, waray pa mga karigo. Talaga, basta waray klase. Ayun. Nagtikada muna ako ng attendees, kaya mag-check na ako ng attendance. Kanina mga nag-leave din ako. Pero, yan. Sige, Ben, kaya mo yan. Padakuha pati yung erong dito. Ay, ito ni Kuna, mata man la ito. Natalata. Uy, Frog, ka-sexy. Akala ko PE. Red bio na klase. Okay, so, mapay skill yun. Mapay yung trap po. Yan, sige. Okay, sige.
Wait lang, sir.
Sige. Ay, ha? Padali na, nawala na akong full screen. Okay, sige, smile ka mo, smile. 1, 2, 3, smile. Charet, charet, charet lang. Okay, so thank you so much, guys. And again, just prepare next week. Direct ganun ka sure kung maray klase. Ikaw pag inampuan nga maray klase kay makuri para makakasulod naman siguro next week. Okay, so kung maray napakiana, thank you so much. And we are dismissed with our online class and hope to see you next week. Bye, everyone.
Thank you, sir.
Yeah, so if you don't have any concerns, pwede na tayong mag-leave sa ating Google Meet. I will send the recorded one after the after mahatag ni Google. Don't worry para ma-receive niyo. Okay, bye everyone.
Thank you, sir.
Bye.